Now that we have examined the mechanisms of primary, junctional, and secondary neurulation, which result in the formation of the neural tube, we will examine how this tube is patterned both along the anterior-posterior axis as well as the dorsal ventral axis. As you can see, a series of bulges becomes apparent in the neural tube at the anterior region, and this actually happens as the posterior portion of the neural tube is still forming. And this is in the chick embryo, by the way, but it's very similar in mammalian embryos as well. Now, these bulges or vesicles will give rise to the forebrain, the midbrain, and the hindbrain. The forebrain, or the prosencephalon, will give rise to several structures, including the cerebrum and the hippocampus, so this region of the brain is important for cognition and memory. The midbrain, or mesencephalon, is important for things like motor control, but also motivation and the regulation of emotions. The hindbrain, or rhombencephalon, develops into the cerebellum, the pons, and the medulla. This region of the brain is involved in the coordination of muscle movement and the regulation of involuntary processes. As you can see, the neural tube goes from these three initial bulges to having more vesicles or more secondary vesicles. The hindbrain, for example, becomes segmented by these swellings that are known as rhombomeres. Each rhombomere is going to produce different neurons as a result of the expression of different transcription factors. Neural crest cells will arise or delaminate from this region of the rhombomeres, and they will form the cranial ganglia or cranial nerves. Part of the reason for the patterning of the hindbrain, so for the fact that different rhombomeres give rise to different cell types, is the expression of Hox genes, along with other factors as well. In addition to the patterning along the anterior-posterior axis, the neural tube also needs to be patterned along the dorsal ventral axis. Different types of neurons can be found in different regions of the spinal cord. For example, the most dorsal region is where sensory inputs are received. The most ventral region contains motor neurons, and between the sensory and the motor neurons, we have interneurons. In order for these different neuronal cell types to be specified, they have to express different sets of transcription factors. The transcription factors that are expressed in each region are in turn determined by morphogen gradients. There are two major morphogen gradients that help to pattern the dorsal ventral axis of the neural tube. These are TGF-beta proteins, mainly BMPs, that are secreted by the epidermal ectoderm, and sonic hedgehog, which is secreted by the notochord. The secretion of these factors from the epidermal ectoderm and from the notochord leads to the creation of new signaling centers within the neural tube, meaning that these factors will eventually be secreted by the cells of the neural tube. Based on the amount of each factor that the cells are exposed to, they will express different transcription factors that will direct them to develop into specific types of neurons. For example, cells that are expressed to high concentrations of sonic hedgehog near the floor plate become motor neurons. Those that are exposed to less sonic hedgehog become interneurons, and those that are exposed to BMP develop into sensory neurons. The role of sonic hedgehog as a morphogen that's important for the patterning of the neural tube has been confirmed by transplantation experiments. If a piece of notochord is placed on the lateral side of a neural tube where it's not normally found, you can see that a new secondary floor plate forms at that lateral side, and that secondary floor plate is flanked by motor neurons. The same result can be obtained if a bead that secretes sonic hedgehog is used instead. So that shows that sonic hedgehog is sufficient to induce the formation of the floor plate and their associated motor neurons. On the other hand, removing the notochord prevents the formation of the floor plate and affects the development of the other cell types that depend on that gradient of sonic hedgehog.